Welcome back to the deep dive. If you're listening to this, I'm guessing you're in what we call the red zone. The exam is probably, a week or two away? Coffee consumption is getting borderline dangerous, and you absolutely do not have time for a 1,000-page biochemistry textbook.
Oh, you definitely don't. And you really don't have time for structural formulas or, drawing out these long, complex pathways. At this stage, it's all about triage.
Exactly.
We have to look at this material strictly through the lens of an MCQ.
So today isn't a lecture. Think of this as the War Room edition. We're taking the biochem rapid revision notes and just stripping them down to the bare metal.
Right.
We want the rate-limiting enzymes, the clinical traps, and those specific numbers that just scream, I'm the right answer on an exam.
Our mission today is to cover carbohydrate metabolism, but we're filtering out anything that doesn't help you solve a clinical vignette. If it's not high yield, we are not talking about it.
Perfect. So let's organize this logically. We're going to follow the timeline of a single glucose molecule from the moment you eat, through fasting, and all the way into starvation.
The metabolic clock. This is the framework for everything. You have to know which fuel is being burned at which hour. It's fundamental.
Okay, so set the scene. It's the fed state. I've just eaten a massive bowl of pasta. My blood sugar spikes. Insulin enters the chat. What's the immediate priority for the body?
Storage and construction. Simple as that. Insulin is an anabolic hormone. It's a builder. It pushes glucose into cells for basically three main purposes.
Okay.
Immediate energy through glycolysis, making NADPH for biosynthesis, and then storing every last bit of the rest as glycogen.
Let's talk about that storage, because this is where the first, numbers question usually hides. The body stores glycogen in the liver and the muscle. How do they compare?
So by concentration, the liver is the absolute champion. You can store about 10% of its weight as glycogen. Muscle is much less dense, maybe 1 to 2%.
But because we have way more muscle mass in our entire body than liver mass.
Right, the total amount of glycogen in the body is actually higher in the muscles. But, and this is the conceptual trap that gets people, the muscle is incredibly selfish.
Selfish is a strong word for biology. Why do we say that?
Because muscle is missing one specific enzyme, glucose 6-phosphatase.
Ah.
Once glucose gets into a muscle cell and gets phosphorylated, it's trapped. It cannot be converted back to free glucose to leave the cell. So the muscle uses that entire stash just for its own contraction. It will never, ever release glucose back into the blood to help out the brain or other organs.
So if a patient is hypoglycemic, their biceps are basically useless to them metabolically.
Completely useless. The liver is the only altruist here. It has glucose 6-phosphatase, so it can break down its glycogen and push free glucose back into the bloodstream to keep you alive.
Okay, let's advance the clock. The pasta is gone. We stop eating. We enter the fasting state. How long does that liver stash actually last?
This is a classic timeline question. For the first, say, 16 to 18 hours, you are running on hepatic glycogenolysis. That's just the liver breaking down its reserves.
And after 18 hours?
The tank runs dry. By roughly 24 to 48 hours, glycogen is pretty much history. The body has to switch gears to gluconeogenesis.
Making new glucose.
Exactly. The liver starts building new glucose from scratch, using things like lactate, amino acids, and glycerol.
And if we push this past 2 days, we're now talking starvation.
Now the whole game changes to spare the glucose. The body shifts to burning fatty acids and producing ketone bodies, and the brain, which usually demands a glucose, essentially agrees to run on hybrid fuel. It starts using ketones to survive.
But there is one cell type that just refuses to adapt, right?
The red blood cells. This is a definitive accept question. RBCs have no mitochondria. They can't use ketones. They can't use fatty acids. They rely 100% on glucose, 204/7.
So even if you haven't eaten in a week, the liver is still churning out a tiny baseline of glucose just for those RBCs.
Precisely. And just remember the hormonal overlords. Insulin owns the fed state. Glucagon owns everything else, fasting and starvation.
Okay, let's zoom in on the engine itself. Glycolysis, this is how we get the energy. But I always see students tripping up on the math here. The net ATP yield, it changes based on oxygen, right?
It's the huge difference between being efficient and inefficient. In aerobic glycolysis, so with oxygen, the net gain is 7 ATP.
Okay, break that 7 down for us.
So you get 2 ATP directly from what's called substrate-level phosphorylation. But you also produce 2 molecules of NADH.
Right.
Since oxygen is there, those NADH molecules go to the electron transport chain. Each NADH gives you about 2.5 ATP. So 2 times 2.5 is 5. Add the original 2 and you get your 7.
Okay, but what if I'm sprinting? Or a tissue is ischemic, anaerobic glycolysis?
The efficiency just collapses. The net gain plummets to just 2 ATP.
Where did the other 5 go?
They were basically lost because the NADH couldn't go to the electron transport chain. Instead, an enzyme called lactate dehydrogenase, or LDH, uses that NADH to convert pyruvate into lactate. You burn the high-energy potential just to keep the cycle spinning.
Speaking of lactate, the notes flagged the Warburg effect.
Yeah. High yield for oncology. Cancer cells they're bizarre. Even when they have plenty of oxygen, they prefer to run on this inefficient anaerobic pathway. They just churn out lactate. It's called metabolic reprogramming.
They're sacrificing energy efficiency for what?
For building blocks, to support their rapid growth.
Got it. Before we leave glycolysis, we need the boss, the rate-limiting enzyme.
It's phosphofructokinase, PFK, or PFK1 to be specific. If PFK is on, you burn sugar. If it's off, you don't. It is that simple.
Now for a practical clinical question from the notes. You're on the ward, you draw blood to check glucose. You have to put it in a specific vial, the gray top. Why gray?
The gray top contains sodium fluoride. See, if you put blood in a plain tube, the RDCs, which we just said are glucose addicts, they'll keep doing glycolysis in the test tube.
Oh, so your lab result will come back falsely low.
Exactly. The blood cells ate the sugar while the tube was waiting to be processed.
So the fluoride just kills the RBCs.
Not exactly. It's more subtle. It inhibits a specific enzyme in glycolysis called enolase. It just freezes the metabolic process so you get an accurate reading of what was actually in the patient's blood at that moment.
While we're on RBCs, they have this unique side hustle called the Rappaport-Lubering cycle. What's the payoff there?
The payoff is a molecule called 2.3 BPG. And this molecule is critical because it shifts the oxygen dissociation curve to the right, which helps unload oxygen into the tissues. But there's a cost.
A cost.
To make it, the RBC has to skip an ATP-generating step in glycolysis.
So they're sacrificing energy to improve oxygen delivery.
Correct. The net ATP for that specific cycle is 0. It's a pure trade-off.
Okay, moving down the line, we've made pyruvate. Now we need to get into the mitochondria. We hit the PDH complex, pyruvate dehydrogenase. This feels like a hotspot for vitamin questions.
It is. It's the cofactor trap.
Yeah.
The PDH complex is very needy. It needs 5 cofactors to work. The mnemonic tender loving care for Nancy is the absolute gold standard here.
Okay, run through them.
T is for thiamine, B1. L for lipoic acid. C for CoA, which comes from vitamin B5. F for FED from B2, and N for NAD from B3.
Basically, if you are deficient in B vitamins, this whole complex just shuts down.
And if PDH shuts down, pyruvate can't turn into a acetyl-CoA. It backs up and it turns into lactate. This is exactly why thiamine deficiency beriberi often presents with lactic acidosis. You see it in chronic alcoholics too.
There's a poison that mimics this.
Arsenic? Arsenic inhibits lipoic acid. So clinically, arsenic poisoning looks exactly like a PDH deficiency. Same bottleneck, same lactic acidosis.
Okay. Once we cross that PDH bridge, we're in the TCA cycle, or the Krebs cycle. I don't want to get bogged down in all the steps. What's the blink and you'll miss it summary?
The yield. 10 ATP per acetyl-CoA molecule. And its regulation is unique. It's not hormonal. The TCA cycle doesn't care about insulin or glucagon.
So what does it care about?
Just the energy needs of the cell. It's regulated allosterically. High ATP, it stops. Low ATP, it runs.
And the poisons. Examiners love poisoning the Krebs cycle.
2 big ones to memorize. Fluoroacetate inhibits aconitase, and malonate inhibits succinate dehydrogenase.
Malonate is the competitive inhibitor, correct?
Yes, exactly. It structurally mimics succinate. The mnemonic from the notes is malasufu. Malonate blocks succinate dehydrogenase, preventing fumarate production.
Malasufu. Got it. Okay, let's step sideways for a second. There's an alternative pathway for glucose, the HMP shunt.
Or the pentose phosphate pathway, yeah.
So here's what always confuses people. This pathway produces 0 ATP, so why does the body even bother with it?
It's not about energy, it's about supplies. It makes 2 critical things, pentose sugars for DNA and RNA, and NADPH. And note the P, this is not the NADH we use for energy.
So what's NADPH for?
Biosynthesis and defense. You need it to build fatty acids and steroids. But for the exam, the defense part is absolutely crucial. In RBCs, NADPH is required to reduce glutathione.
And glutathione is the shield against oxidative stress.
It is the shield. And this connects directly to G6PD deficiency, the most common enzyme deficiency in the world. G6PD is a rate-limiting enzyme for this pathway. If it fails, no NADPH. No NADPH, no reduced glutathione.
And then the RBC is just defenseless.
Exactly. So when a patient takes an oxidative drug like primaquine or eats fava beans, hydrogen peroxide builds up and just destroys the red cell membrane.
Which gives us those classic histology keywords.
Heinz bodies, which are these clumps of denatured hemoglobin inside the cell, and Bike cells, which happen when the spleen tries to literally chew those Heinz bodies out of the cell.
Heinz bodies and Bite cells equal G6PD deficiency. Lock it in.
Okay, but you touch on the other sugars briefly before we hit the storage diseases, galactose and fructose.
Let's do galactose first. There are two types of galactosemia. Which one is the dangerous one?
Classic galactosemia. This is a defect in the enzyme Schulte. Galactose-1-phosphate uridyl transferase. This is the severe one. You see a neonate with cataracts, liver failure, and sepsis, often E. coli sepsis.
And the milder version.
That's a galactokinase deficiency. You mostly just see infantile cataracts, but the child is otherwise pretty healthy. So sick child equals classic GOLD deficiency. Healthy child with cataracts equals kinase deficiency.
What about fructose?
It's a similar pattern. Essential fructosuria is the mild one, a defect in fructokinase. It's totally benign. But hereditary fructose intolerance, that one's the killer.
And what's the enzyme there?
Aldolase B. The baby is totally fine while breastfeeding, but the moment you introduce fruit or juice, anything with sucrose or fructose, they get severe hypoglycemia and vomiting. So the clue is symptoms start after weaning.
Got it. Okay, moving on to storage pathology. Glycogen storage diseases, or GSDs, this is usually a nightmare to memorize. Let's simplify.
Right. We just need to know the enzyme defect and the clinical clue. First, the rules. Building glycogen uses glycogen synthase. Breaking it down uses glycogen phosphorylase.
And remember the hormonal trigger? Insulin dephosphorylates to build. I think of it as insulin makes an incision, glucagon phosphorylates to break, glucagon adds glue.
Nice mnemonic. Okay, the diseases.
Type 1, von Gierke's.
This brings us right back to that selfish muscle concept. In von Gierke's, the liver lacks that release enzyme, glucose 6-phosphatase.
So the liver itself becomes a trap.
Yes. You get severe fasting hypoglycemia because the liver can't release its sugar. So the glucose backs up into all these other pathways, causing lactic acidosis, gout from hyperuricemia, and high lipids.
And the visual cue?
Doll-like faces, these fat cheeks, thin limbs, and a massive liver.
Okay. Type 2, Pomp's disease.
Pomp affects the pump. The defect is lysosomal acid maltase. It's the only one that's a lysosomal storage issue, and it just destroys the heart muscle. You'll see a floppy infant with a massive cardiomegaly.
Type 3, Cory's disease.
Think of this as a milder version of type 1. The defect is the debranching enzyme. You get similar symptoms, liver enlargement, hypoglycemia, but not nearly as severe as von Gierke. A specific clue is the accumulation of something called limit dextrin.
And finally, type V, McArdle's disease.
This is the muscle-specific one. The defect is in muscle phosphorylase. The liver is totally fine, but the muscles just can't access their own glycogen during exercise.
So the patient cramps up immediately.
Classic exercise intolerance. But the diagnostic clincher, the thing they test, is the lactate test. If you make a normal person sprint, their lactate goes way up. In McArdle's, because they can't break down glycogen to make pyruvate, lactate levels do not rise.
No lactate rise equals McArdle's. That's the key. Okay, last big topic. Let's reverse the whole system. Gluconeogenesis, making glucose from scratch.
This happens mainly in the liver and kidney. It's mostly glycolysis in reverse, but you need these special bypass enzymes to get around the one-way streets. The first major hurdle is turning pyruvate back into oxaloacetate.
And the enzyme for that is pyruvate carboxylase.
Which brings us to the ABC mnemonic. It requires ATP, biotin, and CO2.
And the weirdest dietary exam question.
The raw egg white scenario. Sounds made up, but raw eggs contain avidin, which binds very tightly to biotin. If you eat too many, you cause a biotin deficiency. Pyruvate carboxylase fails, gluconeogenesis fails, and you get hypoglycemia.
It sounds like an urban legend, but it's real biochemistry. Last stop, logistics. The glucose transporters, or GLUTs, where do they live?
Okay, rapid fire. GLUT1. Brain and RBCs. High affinity. They're always on, always taking in sugar.
GLUT2.
The sensor. It's in the liver, pancreas, and kidney. The mnemonic is LIPKIS. It has low affinity, which means it only really works when blood sugar is high. This is how the pancreas knows to release insulin after a meal.
And GLUT4.
The most clinically relevant one, adipose tissue and muscle. This is the only one that is insulin dependent. It literally hides inside the cell until insulin comes and knocks on the door.
So in type 1 diabetes, with no insulin, GLG4 never comes to the surface, and the muscle starves even with high blood sugar.
Precisely.
And briefly, GLUT5.
Fructose, F for 5. You find it in the small intestine and sperm.
We should also mention the SGLT transporters, shouldn't we?
Yes, quickly. Sodium glucose link transporters. SGLT1 is in the intestine for absorption. SGLT2 is in the kidney for reabsorption. This one is huge now because SGLT2 inhibitors, the slows-in drugs, are a major class of diabetes drugs. They stop the kidney from reabsorbing sugar, so you just pee it out.
We've covered a ton of ground. Let's do a rapid-fire recap of the rate-limiting enzymes. I'll call the pathway, you call the boss.
Let's go.
Glycolysis.
PFK1.
Glycogen synthesis.
Glycogen synthase.
Glycogen breakdown.
Glycogen phosphorylase.
HMP shunt.
G6PD.
Fatty acid synthesis.
Acetyl-CoA carboxylase.
Gluconeogenesis.
Fructose 1006-bisphosphatase.
And cholesterol synthesis.
HMG-CoA reductase.
That list alone has got to be worth 2 or 3 marks.
Easily.
Before we sign off, give us one final synthesis thought. Why does this all matter? Why is the system designed this specific way?
Well, it all comes back to the red blood cell. It's almost poetic, really. The RBC gives up its mitochondria, so it doesn't consume the very oxygen it's supposed to be carrying.
Right.
It sacrifices its own energy efficiency for the greater good of the whole organism. But that one sacrifice forces the liver to work overtime, creating these incredibly complex pathways like gluconeogenesis just to keep those RBCs alive during starvation. It just shows that metabolism isn't only about chemistry, it's about compromise and survival.
A perfect design with a very high cost. All right, learners, you've got the timeline, the enzymes, and the traps. You're ready for the carbohydrate questions.
Trust the logic. Good luck.
Keep learning, and we'll see you on the next deep dive.